So I'll explain the pipeline so that we can then go into how the trial worked. The PGD pipeline is pretty simple in its design. First, the patients are enrolled. We extract tumor and blood samples from the patients. DNA and RNA is extracted from these samples and sent for sequencing. The sequencing data is then fed into the OpenVax platform, which is Mount Sinai's homegrown neoantigen prediction platform, and it's an open source platform as well. OpenVax helps select the top 10 neoantigen candidates, which are then synthesized into synthetic long peptides. These peptides are assembled into a vaccine at the Vaccine and Cell Therapy Core, also at Mount Sinai, and then administered to the patients in a prime boost format over 10 vaccines spanning a period of 27 to 28 weeks. As an adjuvant, each vaccine also includes poly-ICLC. And throughout the course of the vaccine, starting from pre-treatment to follow-up, we collect blood at regular intervals to perform immune monitoring experiments. So as per protocol, 20 patients were enrolled in the trial. We made the vaccines for 15 patients, and 13 patients were eventually dosed. Of the 13 patients, there were no serious adverse events, so our trial met the primary criteria of safety and also feasibility. Next, we wanted to look at the immunogenicity of the vaccine in the patients. So first, we looked at the inflammation induced throughout the course of the trial in the peripheral blood. For that, we performed Olink analysis on peripheral blood using their Immuno-Oncology panel. And interestingly, we found that each patient had a very unique inflammatory signature, and it didn't significantly change. Minor blips here or there didn't significantly change during the course of the entire trial. Next, we wanted to look at the cellular immunity in these patients to each individual neoantigen in the vaccine. So to look for the de novo immune responses, we performed an ex vivo ELISpot interferon gamma assay. And that assay showed that all of the 13 patients responded to at least one, if not several, neoantigens in their vaccine. So that was very promising and helped us meet our immunogenicity criteria. But then to delve even deeper into the quality of the immune responses induced by the vaccine, we looked at specific CD4 and CD8 T cell responses to either the long epitopes or the minimal epitopes. To do that, we took bulk PBMCs and cultured them in presence of specific epitopes along with adjuvants and cytokines for 10 days. At the end of 10 days, the T cells were re-stimulated with our epitope of interest and then analyzed by intracellular staining and flow cytometry. And the data was actually very interesting. 7 out of 8 patients that we have evaluated so far had a robust CD4 dominant polyfunctional response to several neoepitopes in the vaccines. And this has been reported by other neoantigen trials as well, where while the vaccines are carefully designed to elicit a CD8 response, they do report a much higher CD4 immunogenicity instead. Having said that, though we had a very strong CD4 response, most of the patients also had a polyfunctional CD8 T cell response to several minimal epitopes, which was very promising. So we know now that our prediction algorithm does work. We are able to, and we are constantly tweaking the algorithm. The data from this trial will also go to educate our algorithm further. That we were able to manufacture personalized neoantigen vaccines, administer it to the patients in a timely fashion, and derive an immune response from the vaccine. And remarkably, one of the patients that prior to the trial had progressed on immunotherapy, received all 10 vaccines. As soon as they finished the last vaccine, their disease unfortunately recurred. And then they went on to receive monotherapy pembrolizumab, to which they had a remarkable and clinically significant response and remain disease-free and alive today, which would indicate that our vaccine somehow sensitized the patient to other immunotherapies, and that was very promising, and we are looking forward to delving into that mechanism a little bit deeper. And then also just looking at the T cell responses more qualitatively, identifying the cell states through the course of the trial, and finally validating that the T cell responses we are seeing are actually tumor specific and are cytolytic when they will be co-cultured with the tumor cells.